Welcome to our Knowledge Culture webinar. If you're a Knowledge Culture regular, then welcome back. And if it's your first time with us, then thank you for joining. Today, we're going to be focusing on accelerating your scale-up journey, taking you from Benchtop to commercialization. My name's Stephen Hume from Thermo Fisher Scientific, and I will be your host. Before we look at the agenda today, let me introduce you to our webinar console. So the navigation bar at the bottom of your screen has several options on it. And if you click on any of these buttons, they open and close some of the windows that you have on your screen. And that's so that you can personalize your view of the webinar. So button number 1 is the media player, and that's going to allow you to see our presenters and any video content as well. Button number 2 is the live view of the slides. Button number 3 is the Q&A box. We will be having a Q&A session at the end of the webinar today. So if you have any burning questions, please put them into the Q&A box and our presenters will answer them at the end. And if we can't, we'll endeavour to answer them by email as well. Now, the icon that looks like a question mark is in case you need any technical support during today's webinar. So if anything isn't working for you, then click on the question mark and we'll try our best to help. Of course, we're always interested to hear if you've enjoyed our Knowledge Culture webinars. And so button number 5 is the survey. So you can fill this in at any point of the webinar, and it will also pop up for you automatically at the very end. I'll remind you about it later because we'd be very grateful to hear your comments. The icon that looks like a paperclip is for our resources. We've highlighted some useful resources for you today, and you can access them using this paperclip button. There's also an icon that looks like a person, and that is so that you can request contact with one of our technical experts on one of the topics that you'll hear about today. Now that will send an email straight to me and I will be able to put you in touch with the appropriate person. And finally, the purple box at the end contains information about our Knowledge Culture event happening just next week. And we'd love to see you there too, so check that one out. Now, more than ever, our industry has the opportunity to accelerate the commercialization of biotherapeutics and vaccines. Alongside this opportunity, there are many challenges, and we're going to address 3 of them with you today. Very often, during early phase research and drug development, the focus is on simply meeting the next milestone. Considering process scale-up sooner rather than later can be fundamental. It can improve your time to market, it can ensure data integrity, and it can mitigate additional expenses. So imagine using a consistent bioprocess control software on the latest open architecture controllers, all the way from R&D to production. Or the benefits of using a media panel approach to identify the optimal formulations for your clone. Not forgetting being able to ensure the safety, quality, and efficacy of your product through various analytical detection assays. So with all of that in mind, here's what's on today's agenda. You're first going to hear from Floris Hoyenka, Commercial Sales Leader for the Bioprocessing Equipment and Automation portfolio with Thermo Fisher Scientific. And Floris will be discussing how to select the right bioreactor controller with one of Thermo Fisher Scientific's senior R&D managers, Nephi Jones. We'll then welcome Irina Venk to the webinar. As our process science consultant for cell therapy and cell culture, Irina is going to tell us about accelerating scale-up by using media and feed libraries. And our final presentation will be from another one of my colleagues, field application scientist Susie Brown. Susie will be talking us through Thermo Fisher's rapid solution for Mycoplasma contamination detection. As I mentioned already, we're going to end with our Q&A session, so make sure you put your questions into the Q&A box for our presenters to answer later. Let's find out a little bit more about Thermo Fisher Scientific before we move on to our presentations then. At a glance, we have 75,000 employees worldwide. Our employees are global thought leaders in all areas of science, and we're able to harness the power of that knowledge for you. Our customers are of the highest importance to us. We have 10,000 customer-facing employees so that we're always there for you. 5,000 of our employees are scientists or engineers themselves, so no matter what you specialize in, we have scientists in-house focusing on the same topics. We are, of course, committed to advancing science and innovating, which is why we invest $1 billion in R&D every year. Our mission is to make the world healthier, cleaner, and safer. At our core, we are scientists, and our purpose is to enable our customers to fulfill their scientific purpose. We provide our leading products, services, and workflow solutions using the 6 brands that I'm sure you will recognize here on this slide. And as represented on this slide, the customer is at the heart of everything we do, alongside our focus on innovation, productivity, and quality. Okay, let's get down to business then and introduce our first set of speakers. It's very early in the morning for him as he's joining us all the way from Utah. He holds a bachelor's and master's degree in biological engineering from Utah State University. And with nearly 20 years of experience focusing on feasibility research, product development, modeling, and scale-up, I'd like to introduce you to our Senior R&D Manager of Advanced Technology, Nephi Jones. Hi, Nephi.
Hey, thanks, Stephen, for the intro.
You're very, very welcome. And in conversation today with Nephi is a gentleman who worked in R&D and product development for the first 10 years of his career with the likes of DSM Biologics, Zendo, and Janssen, before moving into commercial teams with Infers Benelux and Finesse Solutions. He joined Thermo Fisher Scientific in 2017, and he's now commercial sales leader for the bioprocessing equipment and automation portfolio in the Americas and EMEA. I give you Floris Hoyenka. Over to you, Floris.
Thanks, Steve, for the nice introduction. Good afternoon, all, and good morning if you're dialed in from a different location like Nephi. I'm very pleased that today we have Nephi Jones attending us for an interview to get more insight about our scaling up from Benchtop to commercialization. Nephi was also one of the founding members of the team that designed the first commercially available single-use stirred tank bioreactor in 2005. So that's really interesting to hear his perspective on that also, as he was involved with the long journey we had on the bioreactor. But first, I want to ask you a poll question. Where in the bioprocessing workflow do you work? Research and development, process development, production or manufacturing, or a supporting role? Give you a few more seconds to answer. See, some people already answered. Thanks for that. Okay, thank you all for who have participated. So within our single-use technology business, we have 5 customer centers located across the globe, which we like to call our customer experience centers, also named CECs. With these locations, we help to address the needs of our customers to see and experience the latest innovations for single-use technologies. Across these locations, we can provide our customers training, testing, application services, and many more. Nephi, thanks for joining us today from your R&D and Customer Experience Center in Logan. May I first ask you to give a brief overview of the work you and your colleagues do at the R&D or CEC department in Logan?
Yeah, sure. Thanks, Floris, for the great intro. Yeah, we have a strong focus on R&D efforts on bioprocessing, right? That's the core of what we do. And our capabilities include process and equipment characterization, process development, engineering scale-up, single-use feasibility, our analytical services, particularly with an emphasis on leachable extractables according to BPOG and training. And we can perform design of experiments that run starting at 1 liters and go all the way to 2K in the legacy sub, and with the addition of 3,000 and 5,000 liters now, terminal scale with the DynaDrive sub. And customers love that we have a truly functional and scalable bioprocess lab, which includes a sandbox for evaluation of single-use mixers and fermenters as well.
Thanks for that overview, Nida. Very interesting to share all the work being done at the Customer Experience Center. So from your experience, what are the key considerations drug developers should think about when considering upstream solutions for early research purposes?
Well, on the assumption that the goal is always to develop a viable drug therapy as fast as possible, then it's typically all about speed. But important is also flexibility in making informed decisions. So speed to get from one scale to the next through a consistent control platform and data management system, and flexibility to adapt and adjust the process as you go to make sure that you're making database decisions. And Thermo Fisher provides this scalable solution through TrueBio. It's a software based on Emerson Delta V platform, and it's matched with our open architecture controllers that can run most brands of single-use bioreactors or even autoclave vessels or fermenters.
Okay, so you mentioned a couple of things here that we can delve into a little bit deeper. But perhaps the first question is, do all customers think about the scale-up journey when selecting their research bench scale technologies?
The simple answer is no. Not all customers, have the same approach. They might have different stages simply because they have different time pressures, or they have, that need for speed, like we mentioned. It creates a lot of pressure to implement solutions, sometimes without thinking through the whole journey. And we find in practice, a lot of times customers, might have existing, sometimes outdated equipment that they're having to utilize early on. And our goal is to really make that scale-up journey seamless and to make the process development, as successful as possible. I like having the picture of the newest subs up here on the slide, the high-performing DynaDrive. I mentioned earlier, innovation is really the heart of what we do, and the DynaDrive is purposely designed to increase the capacity and streamline the end-user ergonomics and meet the scale-up needs of intensified fed batch and perfusion process that's really, intrinsically part of the future of biomanufacturing. And this is a good example of how we work closely, with numerous customers, hands-on, during the early conception, through alpha, beta, into the demo that we've just been doing over this past year, and now the product launch that we're well underway with, this year all the way to 5K. And that's involved bringing customers into the CEC, going to customer sites, and making sure that, their perspective is an intrinsic part of how we approach, process development and how we think of next generation workflow solutions.
I see. So a key point is to really try and think about the whole scale-up journey through as early as possible. And that is something we can help with, of course, because we have already thought through it and have already solutions that can really help with the speed and flexibility of scale-up. So talk about those technologies then. Tell me a bit more about the Bioreactor controllers and the TrueBio software running on Emerson Delta V platform.
Yeah, a key part of being successful is really starting with the end in mind. Right, that's why we have spent thousands of hours characterizing, improving, optimizing the high performance subs. Right, that's now why we offer the 3 and the 5K DynaDrive reactors, is really to allow customers to stay with the benefits of single-use technologies at all scales of manufacturing. Because that platform has an immense and robust design space, it's capable of encompassing both the microreactor, the bench scale reactor for process development, and then also, matching what has been reserved in the past for only stainless steel at 10 and 15K. We can now do that in single use. But like you mentioned, right, it's not enough just to have the best scalability in the industry. We also need to have the automation tools to support them and ultimately bridge research, process development, and manufacturing. And that's really the power and flexibility of TrueBio, is because it runs on Delta V, right? It's the most widely implemented and accepted automation platform in the industry at this time. And we have, right, an open architecture approach. So the benefit is that we can run any bioreactor. We can bring in almost any analytical attribute into the system. And that scales all the way down, right, to the G3 lab. So we've been using that for a number of years. the benefits, right? It's very compact, it's scalable, flexible, it's powerful, and then ultimately it gives us seamless translation across scales. And we're really excited in particular about the GX Core. So that's the latest controller technology. We've just been testing it the last few months here in Logan, putting it through application testing, and it's working beautifully.
Okay, so we can see how the overall package of bioreactors and fermenters, coupled with an industry standard control platform, easy to use software and state-of-the-art controllers can really provide our customers a lot of benefits. But what about the speed and the flexibility they need on their journey from bench scale to commercial product?
Yeah, I mean, now more than ever, right, that flexibility is really paramount. And we have numerous customers that have been successful in scaling past phase 3 into full GMP production campaigns using the G3 Lab and bench scale all the way to the G3 Lite and G3 Pro with the larger subs. Once again, we're especially excited about the DynaDrive. In particular, we've been working with Patheon, a very large and global CDMO, to support a new facility expansion, including the 5K DynaDrive, with the purpose of intensifying fed batch cultures using TrueBio software in that GMP suite, and they're booking clients for the end of this year. So in closing, I want to emphasize the importance of robust data management. After nearly 2 decades of experience doing tech transfer, both large and small scale biopharma companies, I can honestly say that having tools like TrueBio Delta V is incredibly powerful because it reduces the risk profile dramatically and allows for fast and seamless tech transfer to third parties like the CDMO, because you really do need to have that data package to do a good handoff.
Thank you, Niva, for that insight, and I really appreciated that you stood up so early this morning to join us in this webinar. You gave great insights and have given us a journey on the process scale-up and what is important to be successful doing this. As Niva already briefly mentioned, we have launched our next generation controllers, the GX Core. In the following 10 minutes or so, I would like to spend a bit more on this technology and why it's important to choose the right system now that can also fulfill future needs. But first, I want to ask you another poll, which you briefly probably already have seen as the slide was moving, which was not intended to. But here is another poll for you. The question is, do you currently use any of the following bench scale bioreactor controllers? And you may select all that apply. Sartorius Biostat, Appendorf BioFlow, Applicon iControl, MyControl, or Easy Control, Thermo Fisher Scientific G3 Lab or GX Core, or any other. I will give you a couple of minutes or so to answer the question. Give you like 10 more seconds. Thanks for those who already answered. See still some coming in. Thanks for that. Okay, thanks for answering this poll. So, as said, I want to dive a little bit deeper into the GX Core now. So when developing a process in your R&D or process development department, it's quite understandable to focus on the current needs for your bioprocess and to look at the equipment that only fits your bioprocess needs now. However, there are critical points to consider for your bioprocess to eventually lead to a successful bioprocess from R&D to manufacturing. Points are like consistent or predictable product quality, system validation, data integrity, GMP manufacturing, risk mitigation, speed to market. This shows that there are a lot of topics to keep in mind when deciding which bioreactor system suits your bioprocess now, but also in the future. If you only focus on the current research and development process when looking for equipment, you might be tempted to choose the system that is the cheapest and meets your current process needs only. However, this would mean increased cost at the later stage of the bioprocess lifecycle, as other equipment most likely is needed. And this would increase the time to get your drug to market, which again results in an increase in cost. Thermo Fisher offers one hardware and software platform from process development to cGMP commercial production to reduce the complexity involved in scale-up and technology transfer. Recently, Thermo Fisher Scientific has launched the latest member to a bioreactor controller portfolio, the high-performer GX Core. The GX Core controller is built on a highly sophisticated architecture that meets the needs of most bioprocessors in research and development and process development. With the addition of the GX Core, we have a range of bioreactor controllers that can support each step of your bioprocess from R&D up to commercial manufacturing, with keeping the ease of scaling up in mind using our TrueBio software platform. Let's talk a bit more about the unique benefits of the GX Core and how this controller is ideal for your scaling up your bioprocess. The GX Core is an open architecture controller. With that we mean it can run the most of the bench scale vessels up to 15 liter. It can run on high-performing glass bioreactors, but also third-party glass vessels like Sartorius or Apparatus, for example. If you prefer single-use bench scale bioreactors, the system can also run Eppendorf BioBlue single-use vessels. This way you can pick the bench scale vessel that best suits your process. Like all of bioreactor controllers, also the GX Core is running on TrueBio software using the Emerson Delta-V platform. This can be on Delta-V Discovery, specifically for non-GMP processes in R&D and PD, but if preferred, the system can also be running on standard Delta-V if a customer prefers a full Delta-V network and full GMP compliance. As you can see on the picture of the system, the MFC box is placed on top of the GX Core and has 4 MFCs with 2 outlets. The MFCs are set up for air, oxygen, nitrogen, and CO2. Some other benefits are the small footprint and being able to stack the GX Core on top of each other to even further reduce the footprint. Also, the light strip identifies process and alarm state and matches the vessel color assigned in TrueBio for ease of identification of your vessel. At the first glance at this picture, you probably think these are 2 GX Core controllers stacked on top of each other. You could have been right, and stacking 2 GX Core controllers would look similar to this. However, you probably see some differences on the top system, like not having an emergency stop. This is correct, as this is not a second GX Core, but a GX Core expansion model. As indicated before, processes needs change over time during the process development stages. For example, first you only need a bioreactor system to scale up from shaker flask to bench scale stirred bioreactor. The GX Core controller can do this job easily. If you want to further improve your process, for example, into a fat batch with multiple feeds and done automatically, additionally pumps and scales would become beneficial. This is also possible with the GX Core controller, by just adding building blocks to it, like putting 2 Lego blocks on top of each other. And who doesn't like to play with Lego? So the top unit you see here is the GX Core expansion module. So let's explain that in a bit more detail. The high-performance GX Core bioprocess controller is built based on a building block approach, as just mentioned, meeting the basic needs for running a process. As you develop your process over time, expansion modules will be available to facilitate those new needs. On the left of the slide, you see the GX Core controller, which is ideal for bench scale bioreactor glass or single use up to 15 liter. This is a perfect system to start your scale-up journey. As you develop your process further, you might need more capabilities than the basic GX Core controller can offer you. This doesn't mean you can dust ball your GX Core controller and buy a full new system. No, you can add the GX Core expansion pack to your GX Core controller. By doing this, you give the GX Core an upgrade and suddenly have much more capabilities, like for example, an additional 2 built-in pumps, capabilities of connecting multiple scales, multiple spare IOs for connecting any other devices that you require. Quite neat, don't you think? But that's not all. With the expansion pack, you can even do your scale up to pilot scale without the need for a full new controller. The GX Core with expansion module can also run mammalian processor up to 50 liter by using our high-performance single-use bioreactor, but also our latest single-use technology, the 50-liter dyno drive, which Nevi already mentioned. This is a great benefit as this helps you to scale up easily to potential commercial manufacturing scale of 5,000 liter in our high-performance dyno drive. It is also capable to run a high-performance 30-liter single-use fermenter for microbial applications. Additionally, other expansions pack will come available to facilitate high-demanding processes like perfusion. This can be done with the basic GX Core also, but there is getting a higher demand in getting more control on your perfusion process at bench scale size, which is available currently. With the perfusion flow panel, you can have line sets with pressure sensors for inlet, retentate, permeate to monitor your ATF or TFF performance, or flow meters to control the media flow very accurately. All these capabilities will be available with the perfusion flow panel. As you have seen, there will be a lot of capabilities with the GX Core and the expansion packs associated with it. This also requires a consistent software platform that is capable to facilitate all those functionalities without having the need for customizations. This is what TrueBio brings and makes the combination with the GX score so unique and powerful for bench-scale processes. TrueBio is a commercial, off-the-shelf software product that has built-in control strategies, specifically designed for bioprocesses. It is running on the Emerson Delta V platform, which we use for a TrueBio software from R&D to PD to manufacturing. This eases the use of scale-up and scale-down. No programming of Delta V is needed, as the functionalities are built in TrueBio and fully validated. This is really beneficial for the user, as they can immediately start running the process. If needed, they can also easily adjust the settings to their needs, for example, by adding calculation or changing cascade strategies. Eventually, technology transfer to other departments or third parties like CDMOs is also easy, as the data is easily transferable. And as the same software is used from R&D up to manufacturing, limited training is needed. This shows that you can use TrueBio from your initial scale-up from shaker flask to bench scale bioreactor up to full commercial production at 5,000 liter scale using our DynaDrive. So if we summarize the benefits of the GX score, it is an ideal system for companies which do their first scale-up from R&D to bench scale from, for example, a shaker flask. As they develop their process over time, more demanding process and expansion model can be added to facilitate this, for example, for fat batch or perfusion processes. Also, for further scale-up to pilot scale, the GX score controller can be utilized without the need of buying a new bioreactor controller. Ideal for companies that want a bench scale controller that can also facilitate the first pilot process scale-up, like 50-liter cell culture process in the high-performance single-use bioreactor or DynaDrive, or 30-liter microbial cultures in the high-performance single-use fermenter. This shows that there is no need for the customer to replace the bench scale bioreactor system in the coming years when developing their process to the next stage, as they can easily add expansion packages that matches their future process needs. By doing this, CapEx investment is spread across the process development cycle and removes the risk that equipment is bought now but doesn't meet unknown future demands. The final key part is consistency in software and data management. As you scale up from bench scale to full commercial production by using TrueBio software running on the Emerson Delta V platform. With this, Thermo Fisher Scientific has the core solution for scalability by offering a complete portfolio of bioprocess equipment and automation solutions that reduce the complexity involved in scale-up and technology transfer, and enables the data management required for production quality, system validation, and record keeping, from R&D to process development to GMP manufacturing. Finally, I have one other polling question for you, and I want to thank you for your attention. When do you plan to purchase your next bioreactor controller? In the next 6 months? Within 1 year? In more than a year? I'm not sure. Thank you all, and appreciate answering the poll questions so far.
Thank you, Floris, and of course, Nephi as well. That was very interesting, and it gives us a great understanding of what to consider to eventually lead to a successful bioprocess from R&D all the way through to manufacture. And Floris, I must admit, I really loved the Lego analogy as well. That was great. So if you take a couple of seconds to finish Floris's last poll, when do you plan to purchase your next bioreactor controller? And then we will move on to our next presentation. In the meantime, if any of the products that you hear about today are of interest and you'd like to speak further about them, then feel free to click on the Request Contact button in the webinar console, the one that looks like a person. That button will open up an email to me, and I'll be able to pass you on to the most appropriate colleague of mine. So if you'd like to know more about our benchtop bioreactors like G3 Lab or the new GX Core, then we would of course be delighted to hear from you. And just a reminder as well that there is a Q&A session at the end of the webinar. So if you have any questions for Nephi or Floris, then you should put it in the Q&A box. Okay, our next guest has over 10 years of experience in cell culture and upstream processing while working for universities. She joined Thermo Fisher from a CDMO, where she led a project focusing on media optimization and process optimization and development. On the line to us today from Switzerland, please welcome process science consultant for cell culture and cell therapy, Irina Venk. How are you, Irina?
Hey, Stephen. I'm fine. Thank you for the introduction.
Good. You're very welcome.
So then I'll take over here. Good day, everyone. I hope you're doing well at this nice summer sunny day while we are sitting in here that we appreciate a lot. So I would like to talk for the next 10 minutes. I would like to talk about a great process optimization tool you can use shortly before you scale up. But first, I like to change the location of origin of our technologies from the West Coast to the Northeast of the US, with giving you a short introduction to our GIBCO cell culture and cell therapy sites. This map here shows our global GIBCO network. The parent company from which the GIBCO brand was built in the 60s is in Grand Island in the north of New York State. In the last years, we have joined forces with other leading R&D teams from Maryland and Norway. In case you didn't know, GIBCO stands for Grand Island Biologics Company. And if you're curious to know more about this brand, it's always nice to take a stroll on the homepage. You should be able to find it in the paperclip button Stephen mentioned before. What I also would like to highlight are the multiple designated FDA-registered large-scale manufacturing sites across the globe. You will find in Grand Island in the US, Miami, US, Detroit, Paisley in Scotland, Vilnius, Lithuania, and one in New Zealand and Australia. So in this webinar, we have focused on upstream processing scale-up so far, and I would like to highlight now what is important regarding the cell culture medium before scaling up. First of all, assurance of supply is most critical for all of our customers. A medium must be manufactured under certain conditions. A dry format ensures ease of scalability. It needs to be free of animal-derived components. And of course, it needs to be regulatory friendly. So it's actually crucial when testing a medium at large scale or at late scale, not large scale, that it matches all these mentioned requirements. A media optimization tool that meets all the mentioned requirements are actually our media and feed panels that we have developed for CHO and HEK 293 cells. So why would we test a panel instead of one high-performing catalog product to get results quickly? I think the order of the slides is a little bit different than I expected, so I skip this one and go back to that one later, no problem. So as mentioned, why would we test the media panel instead of a very simple high-performing catalog product to get the results more quickly before we scale up? And I think the answer is really simple, and we all know that working with cells, that high-performing cells really need nutritional needs of the cells, have different nutritional needs, because every clone, every cell is unique. Sometimes I don't know the full history of my cell bank, so we cannot predict how they perform best and where they perform best. And this really applies to most cell types used in biomanufacturing. With the picture on the left, I'd like to highlight the uniqueness of HEK 293 cells, what would look comparable for CHO cells. Since the original 293 cells were derived from a human kidney by transformation with adenovirus 5 DNA, a lot happened and multiple variants of it have been created. So on the left, the blue dots on that picture represent adherent cells, whereas the gray dots represent suspension cell lines. So those are on the bottom on the right. Outside of the transformation event, the original HEC is also an adherent cell line being grown with serum supplementation that we don't want to do nowadays anymore. Multiple labs have adopted their HEC 293. We did that as well to grow in suspension, have selected populations that have shown a lower clumping phenotype, adapted to serum-free medium completely, and sometimes even subcloned it. From these historical background and changes, studies have found that various HEK 293 demonstrate a diversity in genome and proteomic profile, what you can see on the right graph. And with all these changes of the original cell line, it's really almost impossible to predict where they would fall in terms of nutritional needs. So the fact of being able to predict in which medium or feed cells perform best led us to develop a media library for basal media, or a library for basal media and feed screening that would increase the chances to find a better fit and improve performance simply. We have developed so far 3 libraries that are available for our customers. 1 base medium and 1 feed panel for CHO cells, and 1 base medium panel for HEK cells that we have just recently launched. You receive ready-to-use bottles of media or feed, so you will not lose any time for ordering process or anything like that. You will afterwards receive the protocols that you can start the test immediately, and of course our technical support to help to focus on your specific process needs and goals. Yeah, and we are talking about acceleration and time. So what's about the time? Scaling up quickly. All this testing will really take very long. And I partly agree. This will take some additional time, for sure. However, I really think it's worth it if you could exceed your performance. So I'm switching back to the slide you have already seen. I hope you can see it now, the media screening timelines. If not, please, Steven, let me know. So this scheme here compares the traditional catalog media approach on the top to the GIBCO media panel solution below. With the catalog option, you can screen catalog products within 2 months, plus 2 months are added for GMP material production, and you can start your clinical trial production process. If you like to add the desired variety to your process by using a media or feed panel option, you will reach manufacturing goals 2 months later. And I mentioned that it will take a little bit more time, but think about the 2 months, it's not really a lot of time. However, I mean, these additional 2 months will give you a very much high probability of better performance. So on the next slide, I'm going to explain you the diversity of the panel a little bit more using the newly launched AAV HEC media panel. So this panel has been developed from over 60 potential candidates to 5 formulations using the so-called principal component analysis, meaning we compared levels of each nutrient in all these formulations 1 by 1. And their effect has been ranked by an algorithm. So 2 main components were used as axes for the graphic on the left. It is to help us to visualize how close these formulations are from one another. And it may seem like panel 2 and 4 are very close to F17, for example. But if you look on the right, at the panel heat map, you will have a better sense on how they differ from one another. This is also demonstrated by the results I will present afterwards. And I'd like to highlight here again that each of the panel, I would like to highlight here, I might have highlighted it already in some of our conversations before, that each of the panel formulations are different from current catalog offerings. They have been selected to provide a broad range of different nutrients concentrations with very specific raw materials. So they are really different from what we offer as catalog product. In order to develop formulations that can increase viral vector production from whichever the lineage the HEC293 is, we used two different cell lineages. This is quite a busy slide, but I'm going to walk you through. One of those two different lineages has been adapted and banked to chemically defined GIBCO media. The second one on the right in another commercial media standardly used for HEC transfection. But also two different clones within the same lineage, the first and second column, were used to show how clone selection can impact performance. Then the three different processes were selected with varying optimal seeding densities. All experiments have been performed independently at least 3 times in shake flasks, and we checked our titers using qPCR at harvest. Our transfection parameters have been pre-optimized for each cell line and selected different pi and DNA ratios for each medium. And maybe that's something I'd like to highlight here. We have used pi and not any other catalog media available on the catalog transfection reagent available on the market to really match our customers' needs. It's also important to underline here, we looked at the effect of basal complex media only. We really wanted to see the effect of this single parameter on the cell line productivity. So in none of the experiments we were using small molecule addition as titer enhancers. So keep everything in mind because I'm going to move over to the results. These graphs show viable cell densities pre and post-transfection. And that data already proves the high variability I mentioned between the 3 tested cell lines and how differently they respond to the different panel candidates. That's quite obvious. Post-transfection viability was high for all conditions with different growth profile in each media. However, there's no correlation between decreased VCD and titers reached. So for this slide here, and that's the very interesting one about the productivities and the titers, I would like to highlight or add one note first. We have normalized all titers to our freestyle F17 medium to compare results better. And the 3 charts show that the 5 panel formulations we have selected from the originally 60 formulations perform very differently depending on the HEC293 cell line. So on the left, for 293F1, we managed to increase the titers by 10-fold. The middle graph shows that we managed to increase titers by 2-fold over freestyle F17 using panel 4 and 5. And for 293S, freestyle 17 titers were similar to panel 4 and 5. I'd like to highlight here that the different panel candidates could have the potential to impact product quality that we have not mentioned here at all yet, as we focused on AAV production so far. But especially for CHO cells, it's crucial to observe titer and product quality. And then it would be beneficial to look at panel candidates with lower titers, which might have an effect on quality. So on the left graph, you see 2 panel candidates, for example, 1 and 3, performing also very well. And you might think about selecting those as well for further experiments. And if I may rewind back to our problem statement that we have high variety nutritional needs and not much time before scale up, why we tend to skip a time-intensive screening. I'd like to remind you that the GIBCO media panel is a screening tool, media optimization tool, that is really ready to be used. It can be easily customized even further, and can be transferred to GMP manufacturing quickly. So it's not a lot of time you have to spend. Panels are really the perfect compromise between working with a catalog formulation that might not completely meet your cell line growth requirements and developing an entire formulation from scratch for your specific process. So it's, as mentioned, a ready-to-go library of unique media and or feeds you get to optimize with personalized support of our field application specialists and R&D team. You can quickly further customize it according to even higher specific needs and can be produced at large manufacturing scale in one of our FDA-registered facilities. So with this, I would like to thank you very much for your interest and hand over back to Stephen.
Thank you, Irina. Thank you very much for your time and for your presentation today. So our final speaker is part of our pharma analytics team here at Thermo Fisher Scientific. And next week, I'll be speaking to our colleague in our Knowledge Culture Spotlight event about simplifying genotypic identification. So a spotlight is a shorter webinar. It's only 15 minutes long, and we thought we'd give you an idea of what that might look like. So here's a quick video about our Knowledge Culture Spotlight next week. So if that looks like it might be of interest to you, you can register for that event on the 27th of July by clicking on the Knowledge Culture Spotlight link in the purple box on your screen. But back to today's presentation then. Our final speaker previously worked as an analytical development scientist with experience in qPCR, Sanger sequencing, and impurity testing. She joined Thermo Fisher 4 years ago and is the subject matter expert in the EMEA region for Microseek. So here to discuss implementation of Microseek and ViralSeek, rapid real-time PCR assays for in-process contaminant testing, field applications specialist Susie Brown. Hi, Susie.
Hi, Stephen. Thank you for the introduction, and hello to everybody on the call. So yeah, as Stephen said, this is what I'll be talking to you today. I'm part of the Pharma Analytics team at Thermo Fisher Scientific, and we offer a set of solutions to help our biomanufacturing customers confirm product safety and product quality. All of our solutions facilitate one of these two highly advanced well-established core techniques shown on the slide. Today, I will be presenting our analytical solutions for Mycoplasma and virus detection, which utilize real-time PCR. Contamination of cell cultures by Mycoplasma and adventitious agents can be very hard to detect and potentially have a devastating impact on a manufacturing site. Traditional culture-based tests for these contaminants have very long testing cycles and high costs, which make them unsuitable for routine contamination monitoring. However, as the industry has been adopting rapid nucleic acid-based methods, which allow same-day results, these assays can now implement in-process testing as a key risk mitigation strategy. The solutions I'm going to cover today are the Microseek Mycoplasma detection kit and the Bioreactor qPCR assays for mouse minute virus and vesivirus detection. MMV and vesivirus are part of the adventitious agent panel that's required for lot release testing of all cell-based products. And these are the two most commonly identified contaminants in mammalian cell lines. Traditional testing points for all three of these assays are at the cell bank stage, at the start of upstream, and then again at the harvest stage prior to moving into downstream purification. But with the implementation of rapid PCR methods, we can now run in-process testing at all stages of your cell culture expansion. So this means that with same-day results, you could detect a possible contamination as soon as it's happened. It enables you to act quickly to minimize the spread of that contamination and stop further wasted raw materials and product from being produced, so thus saving time and money. The Microseek and Bioreactor detection system is a fully integrated real-time PCR-based system. They've been specifically designed to support in-process testing, and additionally, the Microseek method has been further developed to meet regulatory expectations for lot release testing. I'm going to take you through all of the workflow now. So first, I'll go through the Microseek assay. This kit sits as part of this complete end-to-end solution for same-day testing. Here is the overview. First, we start with nucleic acid extraction. That is using our magnetic bead-based PrepSeq chemistry. Extractions can be performed either manually, or we can use an automated express instrument, which is pictured here on the left. This is for fully hands-free processing of up to 13 samples at a time. Next, we have our real-time PCR assay itself. So this is the Microseek kit using cyber green detection dye. It has a multi-primer approach, which allows us to detect over 90 different species of Mycoplasma, and its sensitivity has been demonstrated by internal and external validation to detect fewer than 10 Mycoplasma genome copies, with PCR sensitivity as low as 1 to 3 genome copies. Our latest recommended instrument shown next is the QuantStudio 5 PCR system, and this also comes with dedicated accuracy software that can allow automated presence-absence call for Mycoplasma testing, as well as security, audit, and e-signature features to enable 21 CFR Part 11 compliance. So the full workflow enables you to deliver results in less than 5 hours. As the Microseek assay is using cybergreen detection, we can draw on 3 objective analysis parameters. So cycle threshold, to show the amount of target DNA present in your sample. And then derivative value and melting temperature can be determined by melt curve analysis. And this shows the size and amount of target amplicon that you're producing. So these 3 criteria, which would be established by our customers during their validation, must then be met for a sample to be considered positive for the presence of Mycoplasma. And just to highlight here, because we're using a multi-primer PCR approach, it means that outside of your controls, you only need 1 PCR reaction per sample. This slide quickly shows you a partial inclusion panel for those 90 different species. So the kit can detect over 90 Mycoplasma, Kiloplasma, and Spiroplasma species, and that's with no cross-reactivity, to closely related bacteria. And we have actually directly wet lab tested 46 different species, and they include all the ones commonly used in validation studies, which are those highlighted in red on the slide. And all of those were detected by the assay. The other species up to the 90 have been determined bioinformatically. Another real key point to the Microseek kit is the fact that it comes with a discriminatory positive control. So the discriminatory positive control, or DPC, facilitates amplification of a modified PCR product that has a melting temperature well outside of the real Mycoplasma amplicon range. So when we use post-PCR melting analysis, we will always be able to tell whether a sample contains real Mycoplasma compared to one that contains your positive control. So at the same time, the DPC is maintaining the same extraction behavior as a genuine Mycoplasma DNA, and we use it as a simple extraction positive control and spike directly into our test samples at the beginning. And we can do this without the risk of unrecognizable cross-contamination. Furthermore, we use the DPC as an inhibition control. So at the PCR level, we spike to determine whether a sample matrix has any impact on the PCR itself. Therefore, the DPC is removing the possibility of both a false positive or a false negative Mycoplasma result. To summarize Microseek, it is a real-time PCR assay detecting down to 1 to 3 genome copies of Mycoplasma. We can detect over 90 different species with no cross-reactivity to closely related bacteria. The results use 3 objective criteria. And we have a unique discriminatory positive control that can be used to confirm both extraction and PCR performance without the risk of false positive due to accidental cross-contamination. And finally, the level of support that we provide from Thermo Fisher Scientific means that we're confident that we can enable our customers to progress through implementation, optimization, and potential validation for this assay. So moving across to the Viral-seq assays now. So you can see that these kits sit within exactly the same end-to-end solution. And in fact, you can perform the same upstream nucleic acid extraction on your samples using the PrepSeq chemistry and our 3-in-1 protocol, and then test for all 3 contaminants from just one extraction. And that's because the PrepSeq kit is able to extract total nucleic acid, so we're able to extract RNA and DNA there. The 3-in-1 protocol for Microseek, MMV, and Vesivirus can be performed manually, but it can also be performed on the Automate Express system shown. So for the real-time PCR assays for Viral-seq, we use TaqMan-based technology here, and we're detecting either the mouse minute virus or the RNA virus Vesivirus. These assays, again, can be run on the recommended QuantStudio 5 with the specifically developed AccuSeq software. And again, this is the same less than 5-hour workflow for time to results. The Viral-seq assays have been specifically designed for in-process testing, and they're to detect those key mammalian cell contaminants, MMV and Vesivirus. And we've shown a lower limit of detection down to 10 genome copies per PCR reaction for both of these kits. Vesivirus detection assay enables users to isolate and detect Vesivirus 2117 and all other known strains with high sensitivity and specificity using 2 RNA targets in that kit. And the MMV kit is able to detect all 4 known MMV strains, including the 7 genomic sequences. And both kits have no cross-reactivity with non-related DNA. These are commercial off-the-shelf kits, so they contain everything you need for your assay. And just like the Microseek kit, the Viral-seq kits also come with their own discriminatory positive controls, so that you don't have to worry about any accidental cross-contamination. They also include a negative control and an internal positive control. So this slide just very quickly is detailing how the MMV assay has been designed, but it's the same for the Vesivirus kit, with the exception that the Vesivirus kit has 2 viral RNA targets. So firstly, for the MMV target, we have primers and a FAM probe to detect that. We then have our discriminatory positive or extraction control. This has been constructed to have a secondary target detected by a VIC probe, as well as including the MMV FAM target. Therefore, if you have a DPC spiked or control spiked sample, you will display both the VIC and the FAM signals, whereas a true contaminant result, true positive result, would display the FAM signal only. And then finally, that internal positive control used to confirm successful PCR amplification is by an additional DNA target sequence already added to your master mix, and this is detected by the NED probe. So quickly to summarize, the Viroseek, MMV, and Vesivirus assays are sensitive real-time PCR assays that reach a limit of detection of 20 genome copies per reaction, with high specificity to their respective targets and no cross-reactivity. The results use cycle threshold to determine the presence of a viral contamination. The kit comes also with your unique discriminatory control to confirm extraction performance without the risk of false positives. And again, finally, the level of support we provide will enable you to confidently implement these assays for routine contamination monitoring. Thank you.
Thank you very much, Susie. I appreciate we're at time, but there's still plenty of you watching, so we're going to continue to have our Q&A session as well. So if you do have any questions for Susie, then please put them in the Q&A box, and that will be coming up next. We have a quick poll for you to take before our Q&A, though. You're currently participating in a knowledge culture webinar, and we also run knowledge culture seminars, which are bespoke presentations just for your company. So in a seminar, your company can select from a wide range of workflow and solution presentations, and we'll then organise for a team of Thermo Fisher scientific experts to give you the latest insight on your chosen topics. You may want to have a specific technical meeting on an aspect of today's webinar, or maybe you have other workflows where we can help to optimise your process or solve specific challenges. So the question is, would you like us to organise a one-to-one knowledge culture seminar for your company? Yes, about cell culture. Yes, about pharma analytics. Yes, about production chemicals and our services. Or yes, about purification. Yes, about single-use technologies. So would you like us to organise a one-to-one knowledge culture seminar for your company? And it can be about cell culture, pharma analytics, production chemicals and services, purification, single-use technologies, or maybe you don't need a seminar just now, and that's absolutely fine. And one last reminder before our Q&A session, we have our survey, which you can access down the bottom navigation panel, and it will also pop up at the end, and we'd be delighted to hear your feedback. On to our Q&A session then. Floris, I've got our first question for yourself. It's from Jakob, and he's asking, if I buy the GX Core basic system today, but want to add the expansion module in a year from now, do I need any software or firmware updates or changes to facilitate that? And would it require your service support for installation?
Thanks, Jacob, for that excellent question. And the short answer is no. The expansion module for the GX Core is plug and play, and it doesn't require any software or firmware updates. TrueBio already has all modules equipped in it, and even though the systems is not using it, they are available in the background. So if you add expansion modules with scales or additional pumps in the future, only those modules need to be made visible. You can do it yourself, or we can support you with it. So there's no software changes required for that. So it's really plug and play in that sense. Thanks for providing that question.
Thank you, Floris. Okay, Irina, coming to you for this one. It's a question from Chris, and he's asking, what kind of service offerings do you have with your panels? Can you run some of the testing for us or help optimize the medium or feeds for our clone?
Yeah, actually, as mentioned, we support our customers already with the panel, with protocols and our field application specialists. However, we do offer a so-called FAS consulting service that would include spent media analysis of some selected samples to actually further optimize and customize your process at the end.
Okay, Susie, looking to you next. This is a question from Manu, and she's asking, is there any time difference between running the manual and automated sample preparation for Microseek or Viroseek?
So no, in terms of actual time, the manual versus using the Automate Express works out about the same, around that 2-hour time period. The real difference is the analyst hands-on time that changes. So when you're running that manually, that's entirely kind of in your hands. There are a couple of incubation steps where you can take a break. But otherwise, if you're working with the Automate Express, it can take you about 20 minutes, well, depending, 10 to 20 minutes to set up your samples and the plastics on the machine. And then the rest is done by the machine. You just walk away and come back to get your extraction at the end. So that's the real key in terms of time saved there.
Thank you, Susie, and thank you, Manu. Nephi, one for yourself. This is from Bruce, and Bruce is asking, how do you deal with dissolved CO2 when you're using such large bioreactors? Are the systems really scalable to 5,000 litres?
Yeah, that's a great question, and honestly, that's a lesson I learned a long time ago. I know when I was designing the original 1,000-liter sub, that was really my first experience getting above pilot scale. And at the time, I thought, hey, we'll just use this frit because it generates great KLA, and it ended up being a really disaster because we had so much CO2 buildup. So that was one of my early lessons, and since then, we've spent a lot of time building our characterization capability. And once we got to the point where we had some really good methods for doing KLA of oxygen and CO2, we started to understand the physics of what's going on in the system. And ultimately, that's a lot of the change we've made over the last 5 years, particularly around the precision laser drill hole sparge, is about scalability. And all that is translated into the 3K and the 5K. And you notice those systems, right, they're very tall. And that's really atypical, not standard in the industry to have a reactor that tall. And the reason why we can do it is because of the sparges that we have. Because typically, as those reactors get taller, the residence time is going to shift. And inherently, right, there's higher solubility for dissolved CO2 than oxygen. So it tends to stay in the reactor. That's why you have to have, a lot of times, a macro sparger to strip that out. But we kind of take a different approach with drill hole sparge. We designed it based off impeller interaction, the chemistry of the solution, like the surfactants that are in the process, that change bubble size, and then the surface energy of the materials. So like a traditional stainless steel material is going to have a different surface energy than a plastic like we use in the Dynadrive or the other bioreactors. So the short answer is we've got a new design, and ultimately we scale the pore size to match the column height. And we found that if we can stay within a certain entrance gas velocity, that we can avoid the turbulence, we can avoid basically that bubble getting larger or smaller depending on flow rate. And that way, regardless of flow rate, the bubble size is going to be predictable. So we match not just the pore size to the column height, but also the number of pores to keep the cross-section in check, so we can stay in that ideal spot that we're under the shear threshold and the physics that make the bubble creep or coalesce. So really with the enhancements we've made to the sub and the Dynadrive from the get-go, scalability is no longer an issue when it comes to dissolved CO2. And that's really important too, because the challenge is that you're not going to see those issues at small scale. When you're working in amber, you're working at 1 or 2 liter on the bench, you're never going to see dissolved CO2 as a problem if you're running a typical cell culture process, just because the surface to volume ratios of the reactor are completely different than when you get to large scale. So that's, once again, that's kind of why we start with the end in mind, is we want to make sure we do characterization of the terminal scale, understand its limitations, and then make sure the scale down model really matches well to the big picture.
Brilliant, thanks, Nephi. And sticking with yourself for the next question as well, if that's okay. It's from Annabelle, and I do hope I've pronounced your name properly. Deep learning and artificial intelligence is moving quickly into the technology. Do you have any appreciation about this?
Yeah, I mean, it's interesting to see how much that is getting traction in technology. Deep learning, machine learning, artificial intelligence, right? It's definitely prominent in a lot of industries. I think our industry, right, is definitely seeks for innovation around biopharmaceutical, biologic production and discovery, but they're also risk averse. So I think the trend is going to be slow, but it's definitely entering in already on the research scale and in process development. And ultimately, we're working very closely with Emerson. So we're going to leverage their tools, and they see a lot of different industries that I think move a little bit faster than biopharm, so we can apply those best practices. And maybe I'll turn over to Floris. He might have something particular that he wants to mention.
Floris, any thoughts?
And
Sorry,
maybe
I was listening.
this.
It was breaking up. Sorry, I had an issue.
Oh, sorry, Flora. So I was just mentioning that I was going to mention that basically TrueBio 6 is going to be launching here in the next year, and I think our goal eventually is to bring some of those machine learning tools into the next launch eventually.
Yeah, there's things we continuously are updating also with our software, and also to look at process modeling and things like that. So that's definitely something on the road where we're looking at, as we see that in the market there are more and more needs for doing that. So that's definitely something we have on the radar.
Thank you both for that answer. Floris, sticking with yourself then here, you indicated that the GX Core with expansion module can also be used with the pilot scale vessels like a 50-litre single-use bioreactor. Alina is asking, does this mean she has to buy a new controller with that pilot scale vessel, or can she use one of her existing controllers that's also running bench scale vessels?
Yeah, thanks for addressing that question. And that both is possible. If you buy, for example, today 2 GX Core controllers to run 3-litre glass bioreactors, and after a year you want to try your first scale up to 50-litre, for example, you can reuse one of those controllers with running maybe, for example, a 50-litre Dynadrive or a 50-litre High Performa, and keep the other one still running a 3-litre, so you have a satellite run next to each other. So you would only need to buy the expansion model for your GX Cores so it can facilitate that large scale, but you can reuse that existing controller, and you can be as flexible in that sense, choosing the vessels you want to use with your controller. Of course, you can extend it further. Maybe you want to add a third one or a fourth one. That's also possible. So it gives you the flexibility in that sense. But in summary, there's no need to buy an immediate new controller. With the expansion model, you can run the pilot scale also.
That's great news. Thank you, Floris. Susie, can I use other AB instruments for the assays? And would it work on a Quant Studio 7, for instance? That's a double question from Karesh.
So yes, they are either TACMAN or cyber-based assays, so they can run on any qPCR system that you've got. So yes, in the example, the Applied Biosystems Quant Studio 7 would work. The reason we talk about and promote the Quant Studio 5 is that it runs with the dedicated Accuseek software. So that software has really been built for our pharma analytics customers. So we have templates already set up. The instrument is designed to do automated cooling or provide the calculations all within the system. But ultimately, yes, we will support you with whatever workflow you need to implement your kits with. So good question. Thank you.
Thanks, Susie. Floris, coming back to yourself then. Here's a question from Adam. Adam's interested to know, are the Thermo Fisher bioreactor controllers for above 50 litres using the same TrueBio software as the bench scale units, or does it differ in user interface?
Thanks for asking that question, Adam. That's an important one. In summary, they are all running on the same TrueBio software. As mentioned also in my presentation, we have a non-GMP version running on Delphi Discovery, and we have a GMP version running on standard Delphi. Both the same user interface, same functionalities. It's just that the non-GMP version is really a PC-based, so a cheaper solution for non-GMP environments. Our GX Core can run on both, depending on what your needs are. A small network maybe, and PD environment, maybe Discovery will be sufficient. But if you want to have a large TrueBio, also that can be done with GX Core. That same counts for a large-scale equipment that will be running on TrueBio native Delta V for a full production scale, which is also GMP compliant.
Thanks, Floris. We are going to come back to you as well for this one. It's a question from Umit, and they're asking, there are fundamental pricing differences in small-scale reactors, which drives choice. So, Floris, maybe you'd like to discuss that point further?
Yeah, thanks for answering that one. And that's always a discussion we have with customers. And that is always, of course, a balance, the budget that is available. I think I mentioned it also in my presentation. It's not always about money now, but also thinking ahead. So it could be that it's cheaper at this stage, but it will bring you additional cost in the future if you want to go further in your scale-up journey. So that's really the thing to keep something in mind, not only to focus on the now, but also what potentially are your future needs. And I think with the GX score and also with TrueBio, you have a controller that can adapt to future needs. I hope I made that clear in my presentation. But also TrueBio is a software that grows with the journey also with you. We have it from bench scale up to full production. And that is also a reducing cost immensely, because it's not only the CAPEX, but it's also, if you do the tech transfer, it makes the ease of tech transfer also easily, so less money is spent in training the people on that platform if you already have that same platform. I speak from own experience back in the lab that it can be really different if you have different user interfaces in that sense, or platforms to run it, and you go from bench scale to production scale. And in that sense, the GX score is also really beneficial, because even though you maybe have a preference for a third-party vessel, as mentioned, maybe not our glass vessels, but maybe a Sartorius glass vessel or an Applicant glass vessel or an Eppendorf BioBlue, the GX score can run that. So you're quite flexible in that sense, but you will have a consistent user interface, and that will eventually save also a lot of money. So my advice is, not only focus on the now with the investments, and I know that's not always that easy, but also look ahead in the future what your plans are for that, and what changes might come, which you have to take into account.
Thanks, Floris. And we have had a question in which I'm going to take the answer to as well, which is, will this webinar be available on demand? And yes, it will. So within about 24 hours or so, you'll be able to watch the whole webinar back on demand as well. So I hope that helps, Annabelle. Susie, for you, Kelly's asking, how can I tell if there is matrix inhibition in my Microseek assay?
Okay, thank you. So the use of the discriminatory positive control is used to tell that. When we spike at the qPCR stage, we spike as a positive plate control, our positive PCR control. But we also spike that DPC into your test sample itself, so your extracted sample. And then we do a delta CT, so look, we compare your positive control to your positive in your sample, and we should see that they don't differ by more than 2 CTs difference. If we see more than that, then that's a clear indicator that there's some sort of inhibition going on. So we've got that as a control for every test sample that you run. Thank you.
Thanks, Susie. Thank you, Kelly. I appreciate we're almost 20 minutes over time, so I'm going to make this the last question. Floris, it's for you. It's from Anila. Does the type and quality of instrument or apparatus used in production affect the scale-up process?
I think it comes back to the question I just answered before, is having the same user interface across the board will really help. Of course, there's different qualities in that, but I think that really can easily the tech transfer. And with our controller platform, you have the flexibility in using the vessels you want, so you're not stuck to one vessel from bench scale and large scale. So I think the quality is also what you can do with it, and especially data management and how that is handled. And I think with our TrueBio Delta V software, that really is a key point, is the data management and to secure the data. And that is always something to be aware of. Delta V is widely used in the industry, specifically of those reasons, the security of your data, but also there are a lot of things to keep the network secure. So I think that's really a key topic to keep in mind when running your manufacturing process also.
Thank you, Floris, and thank you to all our speakers today for your time and knowledge. We really appreciate it. I'm afraid that is all we have time for today. I do hope you've taken something from today's webinar on accelerating process scale-up. We've looked at how selecting the correct bioreactor hardware and software can set you up for commercialization. We've looked at how GIBCO media and feed panels can accelerate your development. We've looked at how you can implement rapid real-time mycoplasma contamination detection. If you have asked a question that we didn't manage to answer, and there was a couple of them, don't worry, we will endeavour to answer all questions by email. Please do fill out the survey that will pop up when the webinar closes. Thank you very much for your time today. Stay safe and have a great rest of the day. Goodbye.